Alcoholic and gout. Why is it so? Let's uncover all these facts in this. Now, alcohol is metabolized into acetaldehyde with the help of NADH using the enzyme alcohol dehydrogenase, and it gets further oxidized to acetic acid using acetaldehyde dehydrogenase enzyme, where NAD gets reduced to NADH. Now, alcohol dehydrogenase is a cytoplasmic enzyme, whereas acetaldehyde dehydrogenase is a mitochondrial enzyme. Now, this cytoplasmic alcohol dehydrogenase has got high Km value, which means that large amount of ethanol, when it is present, it gets converted to acetaldehyde. Whereas mitochondrial acetaldehyde dehydrogenase has low Km value, which means that when acetaldehyde is present even in low concentration, it gets oxidized to acetic acid. So one thing you need to remember is this mitochondrial acetaldehyde dehydrogenase is less in Indians when compared to Western individuals, and this is the reason why Indians are more prone for chronic ill effects of alcohol, and cirrhosis is more common among Indians. So this is one pathway. The other pathway by which alcohol is metabolized in the body is by microsomal ethanol oxidizing system, which uses NADP to oxidize ethanol to acetaldehyde, and NADP gets reduced to NADPH. This uses cytochrome P450. Further, acetaldehyde gets oxidized to acetic acid by acetaldehyde dehydrogenase enzyme. Now, this cytochrome P450, that is, this is induced by ethanol, and this will definitely impair the drug detoxification and rapidly clears the drug whenever consumed by an in chronic alcoholic individual. Apart from this, ethanol accumulated has got so many ill effects, and these include ethanol inhibits the inhibitor neurotransmitter GABA, and it stimulates the inhibitor neurotransmitter GABA and inhibits the excitatory neurotransmitter, resulting in CNS depression. Ethanol also causes premature activation of the digestive enzyme, resulting in auto-digestion of the asana cells of the pancreas, predisposing to the development of chronic pancreatitis. Ethanol combines with fatty acids to form fatty acid ethyl ester, and this will also damage the pancreatic asana cells, predisposing the individual to the development of pancreatitis. Apart from that, ethanol also stimulates reactive oxygen species, resulting in apoptosis of different cells of different organs. So the other pathway is where the ethanol gets oxidized to acetaldehyde, and this is associated with increased generation of NADH. Now this will facilitate conversion of pyruvate to lactate, and lactic acidosis sets in, and this results in the development of high anion gap metabolic acidosis. Lactate competes with uric acid for excretion by the renal tubules, and this predisposes these individuals to the development of hyperuricemia followed by gout. Now, this NADH, increased formation of NADH, facilitate conversion of oxaloacetate to malate. So, as there is no oxaloacetate available, so will be no gluconeogenesis, and alcoholics are prone for development of hypoglycemia. Since the oxaloacetate is deficient, a TCA cycle will not operate, and the acetyl-CoA gets diverted to form fatty acids. So, increased fatty acid formation predisposes the individual to the development of fatty liver, and over a period of many years, fatty liver progresses to development of cirrhosis. Increased acetyl-CoA is also a key starting material for the formation of increased cholesterol. So there occurs increased formation of total cholesterol, increase LDL cholesterol, decrease HDL cholesterol, resulting in dyslipidemia, and over a period of many years predisposes these individuals to the development of atherosclerosis. So all these are because of increased NADH generated in the body. Apart from that, acetaldehyde itself is a toxic biomolecule, and this can inhibit pyridoxine, resulting in development of peripheral neuritis. It can inhibit thiamine, predisposing these individuals to the development of cerebellar ataxia, nystagmus, psychosis, manifesting as Wernicke-Korsakoff psychosis. It inhibits, acetaldehyde inhibits the asana cells of pancreas, resulting in development of pancreatitis. It also inhibits the incorporation of sialic acid, also called as N-acetyl neuraminic acid, into transferrin. This desialated transferrin is used as a marker of alcohol abuse. Now, this chronic alcoholism, the addiction is overcome by administration of a drug called disulfiram. So, when this disulfiram drug is introduced, so what happens? There is This drug inhibits the enzyme acetaldehyde dehydrogenase, so acetaldehyde accumulates, and this is also responsible for alcohol flush reaction, which includes nausea, vomiting, facial flushing, palpitation, the hangover symptoms. So this was about alcohol metabolism. Now there is a difference between the manifestations of acute alcohol intoxication and chronic alcoholism. So in acute alcohol intoxication, when a person consumes 5 milligram per deciliter of alcohol, the person is normal with all the normal functioning. So with consumption of 50 milligram per deciliter, the person shifts to a state of euphoria, elevated state. With consumption of 150 milligram per deciliter of alcohol, neuromuscular incoordination sets in, characterized by difficulty in driving. With 300 milligram per deciliter of alcohol consumption, the speech is affected in the form of slurred speech. With 400 milligram per deciliter consumption, stuporous stage is attained, and with 600 milligram per deciliter, coma sets in. Chronic alcoholism, so the liver is affected with fatty liver, which progresses to cirrhosis if alcohol consumption is not stopped. CNS effects are characterized by depression because of stimulation of the inhibitor neurotransmitter. So one is Korsakoff psychosis due to inhibition of thiamine, and peripheral neuritis due to inhibition of pyridoxal phosphate or pyridoxamine. So cardiovascular system effects include because of excess cholesterol accumulation predisposing to the development of dyslipidemia and atherosclerosis, impaired drug detoxification due to induction of cytochrome P450 with rapid clearance of drugs, hemolytic anemia, the RBC membrane is altered because of increased cholesterol, and anemia, hemolytic anemia sets in. So the laboratory findings in chronic alcoholism individual includes increased amylase and lipase which are released from the damaged pancreas, increased gamma glutamyl transferase and increased desialated transferrin are marker of alcohol abuse, increased alanine transaminase and aspartate transaminase are released from the damaged hepatocytes. Apart from that, because the hepatocytes are damaged, the protein biosynthesis is decreased, so serum total protein and all decreases. So this was all about alcohol metabolism. Happy learning. Next video will be on CSFs. Please do like, share and subscribe if you are like this concept. Thank you.